Hi, everyone, and welcome to this next session. My name is Allie Osgood. I'm a program analyst at the NHGRI, the National Human Genome Research Institute, and I believe Ruth Chadwick will be joining us to chair soon. But in the meantime, we will get started. And our first presenter is Professor Arisa Mwaka Sabakaki, who will be talking about perceptions and experiences of researchers and research nurses on the informed consent processes for genomics and biobank research. Over to him.
Good morning, ladies and gentlemen. My presentation on the perceptions and experiences of researchers on informed consent process for the informed consent process for genetic research. I'm Associate Professor Mwaka Arisa from Makerere University. That's the list of my co-investigators. The aim of the study was to explore the perceptions and experiences of researchers on the informed consent process for genetics and biobank research. We used a convergent mix and 15 nurses and study clinicians. The majority of the participants or respondents in the survey were male, and 85% of them were involved in scientific research, and 34% were in academia. That means they are university lecturers or professors. 86% of them had collected samples for research before, and 89% were involved in collaborative partnerships or international collaborative research. Respondents were asked for their opinion on what should be in an informed consent form for sample storage. And basically, the majority of them indicated that informed consent forms should provide donors with the option to consent to future research. And for those ones, and for the consent form, those ones with options for use of samples in the future, consent forms that give donors the option to consent to future research should include multiple options regarding the types and conditions of the future research for which the samples may be used. Now, this is in line with broad consent, and 62% indicated that broad consent was acceptable. However, 38% included that two options can also be included, either to consent or not to consent to all future research. Now, this one is in line with the blanket consent, which is not permissible under the Uganda national guidelines. Now, when it comes to the use of consent for sample storage, the majority of participants or respondents indicated that this should only be used if they are anonymous and all linking information has been removed. Others were of the view that donors should only be able to provide consent for research on their samples if they have the opportunity to withdraw consent, and if the donor will be notified if any information relevant to the donor's health is discovered. However, there were varying opinions when it came to limitations to the use of samples. So some participants were in agreement, others did not agree with there being any limitation. Some of them indicated that there should be no limitation on the advance of consent that donors may provide as long as future research on the samples is approved by the Research Ethics Committee. On the issue of reconsent, the requirement for reconsent before using donor samples in research, the majority of participants indicated that it would place an unacceptable burden on researchers and would be prohibitively costly. However, there were varying opinions when it came to the inconvenience to donors. You find that some members were not in agreement with the inconvenience to donors, whereas others thought that it would be an inconvenience. So we went ahead and conducted qualitative in-depth interviews to really try to delve deeper into what we had obtained in the survey. And basically, data were collected under five thematic areas: general understanding of genetic research and consent options for sample storage, information disclosure, ethical, legal, and societal issues in informed consent, challenges in informed consent processes for genetic research, and recommendations for enhancing comprehension of consent. Those were the demographics of the participants. Under the general understanding, the majority of research participants did not understand what broad consent is. A case in point is a quote from one of the a geneticist, and I'll quote, "But this is consent that is offered by the participant without any restrictions in the terms of use for the samples." The question that you need to ask is, should the participant restrict this in terms of time? I donate my samples, should it be used forever or restrict it to a particular? Now, this is a description of blanket consent and not broad consent. And the majority of PIs were not well conversant with the informed consent procedures of their studies. This process was delegated to the nurses, nurse counselors, and study coordinators. And most nurses lack basic knowledge and understanding of genetics. And these are the people who are supposed to ensure that participants understand genetics. Several benefits of genetic research to humanity were highlighted, and all researchers and nurses concurred that there are no immediate direct benefits to participants. However, clinically relevant findings can be of direct benefit to individuals, their families, and the community. And several risks were also highlighted. For example, breach of confidentiality, social harms, economic harms, then the implications of the return of results, that's primary results and incidental findings to individuals, their families, and the communities. This is a description of blanket consent and not broad consent. And the majority of PIs were not well conversant with the informed consent procedures of their studies. This process was delegated to the nurses, nurse counselors, and study coordinators. And most nurses lack basic knowledge and understanding of genetics. And these are the people who are supposed to ensure that participants understand genetics. Several benefits of genetic research to humanity were highlighted, and all researchers and nurses concurred that there are no immediate direct benefits to participants. However, clinically relevant findings can be of direct benefit to individuals, their families, and the community. And several risks were also highlighted. For example, breach of confidentiality, social harms, economic harms, then the implications of the return of results, that's primary primary results and incidental findings to individuals, their families, and the communities. We also ask questions on information disclosure during the informed consent processes. And informed consent processes should use an iterative approach that involves several rounds of to and fro communication, encourage consultation with family and other people, participants trust, the use of simple language and use of visual aids and other medias to try and ensure that participants understand, at the end of which there is an assessment of comprehension and finally authorization. It was also noted that it's very important to use informed consent forms translated in the language that the participant understands well. However, as we'll see later, there are quite a number of challenges when it comes to translation of scientific terminology into local languages. Participants also indicated that some of them use peer educators and peer group discussions for enhancing participant education. There are research participants who understand much better than others. Then there are others who have participated in genetic research, and others actually also try to help the community board, community advisory boards in community engagement. And these ones help in peer education. Community engagement was also seen as key in the education and sensitization of community. There is also need to respect local culture and beliefs and dispelling of misconceptions. For example, this is a quote from a researcher. People have certain beliefs they may believe that you are going to use the blood for other purposes. They may think that it is either witchcraft and so on. And this is where community engagement comes in. It's very, very important that these misperceptions are identified and dispelled. Participants also thought that participant concerns and preferences should be respected, and assurances for protecting privacy and confidentiality should be given. On the ethical, legal, and social implications of genetic research in informed consent processes, participants noted that differences in regulation and guidance in collaborative research at times cause problems during the informed consent processes. A case in point is the definition of human participant research in the US vis-a-vis in Uganda, whereby anything that comes out of the human body, whether it's anonymized or not, is considered human participant research. There were also fears of loss of control and inability to monitor the use of exported samples. And these are fears, and they are fears of misuse and exploitation. And these fears are both for the researchers, and they also indicated that participants also asked questions about the misuse and exploitation of their samples. And then actually the researchers indicated that there's some negative attitude towards the export of samples from research participants. And some research participants actually withdraw from studies or refuse to consent for studies because of export of samples. Other issues that were identified were issues on benefit sharing, sample ownership and commercialization of samples, and data sharing. Societal implications, social harms, and I'll quote here, yes, it can affect a family in case you disclose findings unnecessarily, maybe to the wrong people, or in a way that is not right. It can affect a family. It can even bring a breakup in a family and issues in homes and children. So it's very important. It's a very important issue and sensitive in the community. There are also issues of stigmatization and discrimination. Some researchers also raise the issue of biosecurity, whereby people can misuse the data and the samples for their own ends, and in the end up causing national security problems. processes, procedures, and implications for research, return of results and incidental findings. Right now, most studies in Uganda are not returning these results. However, researchers do not have any particular guidance on how this should be handled. H3 Africa has come up with a guidance document, but this is unknown to most researchers in Uganda, and actually we do not have national guidelines on the return of genetic results. Quite a number of challenges were also identified, and this one, the most challenging is the poor quality and inaccuracy of translation of consent forms into local languages, inadequate understanding of consent by participants due to illiteracy, high literacy levels, use of multiple informed consent forms, lengthy consent procedures that affect participant concentration, voluminous informed consent forms, etc. There is also limited understanding of genetics by communities and some research team members, especially the nurses and nurse counselors who obtain the consent and are supposed to make sure that participants understand. Then there is also lack of professional genetic counseling services. These are key in especially when it comes to return of results and explaining to participants what the implications of these genetic results are. Then there is also mistrust of foreign collaborators. Several recommendations came out of our data, recommendations to enhance comprehension of informed consent. Community engagement was seen as key in sensitizing the general public and educating them on genetic research and its implications. Researchers also encourage the use of iterative approaches to informed consent, where participants are given ample time to read or consent forms to be read to them, and the opportunity to ask questions, make consultations with families and trusted ones. Then also encourage the use of simple languages, simple local languages, and various media during information disclosure to ensure that participants really understand the information that they are given. There is need for harmonization of translation, a dictionary of translated key scientific and medical terms or concepts used in research and clinical care in local languages should be developed. Then the national guidelines specific for the conduct of genetic research should be developed, or the current national guidelines should be amended to include a specific chapter on genetic research. And research ethics committees should be trained in the basics of genetic research in order to ensure that they appreciate the ethical, legal, and social implications of genetic research and are competent enough to review genetic research. The use of checklists, right now in our national guidelines, the use of checklists in assessing for comprehension is not mandatory. However, participants have the view that the use of checklists for assessing understanding of consent should become mandatory and should also be included in the national guidelines. And the Ministry of Health and Research and academic institutions should be encouraged to build capacity for genetic counseling, because this cadre of professionals is non-existent in the public. In our public service positions, and this should be encouraged since genomic research is on the increase. Thanks very much for listening to me. Questions are very welcome. Thanks very much.
Thank you for that talk. Just want to remind all attendees that you can ask questions in the live Q&A chat, and please make sure to address your questions to the speaker you would like to answer that question. And after these next two speakers, then we'll have the Q&A session. And up next, we have Ms. Dimfo Rala, who will be talking about expectations and preferences for of individual genetic results. focusing on participants' views from an HIV TB genomic research project in Botswana. We will turn it over to her. Thank you.
Good day, everyone. My name is Dimpara Lefala, and I'm a PhD fellow at the University of Cape Town. I'm going to present on expectations and preferences for feedback of individual genetic results, participants' views from an HIV-TB genomic research project in Botswana. As you might all be aware, whole genome sequencing sometimes reveal information about individuals that is unrelated to the original research question, and these findings may be important for patient care. For example, researchers may identify that a participant carries a mutation that predisposes them to developing a particular illness or that influences their drug metabolism. These results may be clinically actionable, unactionable, or without significance. While most studies conducted in high-income contexts reveal that participants generally would like to be provided with the feedback of their individual genetic results, such work has hardly been extended to low middle-income contexts, particularly in Africa. Therefore, it remains unclear which individual genetic results African participants would like to receive, how and when. As a result, we conducted an empirical study to investigate questions related to what genetic results people may want to know, how they want to know such results, how they would like to receive such results, and when they would like to receive such results. We conducted a qualitative study employing deliberative focus group discussions as well as in-depth interviews. And we specifically targeted parents and adolescents enrolled in a conversion study in Botswana. Overall, we enrolled a total of 93 participants. 49 of these were parents and caregivers, and 44 were adolescents. We held altogether a total of 12 DFGD meetings with the parents and caregivers, as well as 12 DFGD meetings with the adolescents. We additionally held 6 in-depth interviews with parents and caregivers and 6 in-depth interviews with adolescents. To explore participants' preferences for the feedback of results, we gave them a table that was extracted from Home et al. 2015, which categorizes genetic results into 4 categories. We found that parents and caregivers wanted to receive results across all 4 categories of genetic conditions discussed, while adolescents were reluctant to receive results for genetic conditions that were severe, non-preventable, and unactionable. We explored participants' reasons for wanting to receive genetic results, and participants indicated that it is important to know the genetic conditions that they are predisposed to. They shared that this knowledge will help them to be mentally prepared and accept their situation. They also shared that the knowledge of their genetic condition will help them to improve their lifestyle. They will be able to do exercises, for example, and adopt healthy diets so that they can prevent the progression of a preventable genetically predisposed condition. They also shared that knowledge of their genetic results will help them to look for help, look for treatment where possible, or look for other ways of managing the genetic condition. They additionally shared that this knowledge will help them to prepare for the future. They will be alert about their health and prepare for whatever will come their way in the future with regards to their health. Lastly, they shared that they would like to receive their genetic results because they would want to share them with their family members, because these results might also be important to their family members. We identified key areas for consideration in the interpretation of participants' preferences for feedback of findings. Firstly, participants' context. It seems that the choice of which results people might want to receive is influenced by their experiences. For example, parents and caregivers in our study wanted to receive all genetic results, while adolescents were reluctant to receive genetic results for conditions that are severe, not preventable, and unactionable. This may be because parents survived a time where HIV was not treatable, there was no treatment for HIV, and are now living at a time where HIV has treatment and there are several ways of managing it. And these experiences seem to have influenced their views on receiving genetic results despite their severity, preventability, and actionability. This observation is similar to those of parents of children with rare diseases. In Klederman's study, for example, parents preferred to receive all types of results of their children and ascribed this to their success in coping with their child's condition. Secondly, the participants' relation. Participants valued the importance of sharing information with family members. They seemed to view their lives to be interconnected with others, especially family members. This view is consistent with the African way of life, which is communal and where there is a strong emphasis on solidarity. Thirdly, empowerment. Participants attributed a lot of power to knowledge. They wanted to know their genetic results for the sake of knowledge and considered knowledge about their genetic predisposition will empower them to act in favor of their health or to help those around them. Participants were also investigated on how they would want to receive their genetic results and whom should feedback these results. Almost all the participants would prefer to have their results fed back in person. Most adolescents would prefer to have these results fed back by the researchers, while parents and caregivers would rather have researchers work with the doctors to relate the results to participants. Both adolescents and parents expressed that feedback should be supported by counseling, but we would not inquire whether this counseling should be done by genetic counselors. In conclusion, most perspectives suggest that the participants wanted to receive their genetic information, and this varied based on severity, preventability, and actionability of results. As a result, there seems to be a stronger case to have actionable genetic results returned to participants regardless of age, as this information was highly desirable. Lastly, decisions on practicalities for feedback of results should take into account participants' context and preferences. Thank you.
Thank you very much, Dinfo. We'll go straight on to the next talk, which is on a related theme by Mayara Fontes Marx.
Hi, everyone. Okay, let's start. Hi, everyone. My name is Dr. Fontes Marx from the Infoengenharia Group, and I'm here to present two of our studies. The first one is a systematic review of the economic evaluation of returns IF in genomic research, and the second one is the cost of returning Africa-recommended IFs to research participants, a cost framework. So a little bit of, there is a lot of discussions regards who and how IF should be returned, and the ethics behind returnings have been increasing dramatically over the years. Information on the costs and benefits of returns IF to participants remain very scarce. Thus, the objective of this study. This study was to systematically review all the economic evaluations of returns IF studies in the genomic research. So the method was we look for published articles on the cost effectiveness, cost benefits, cost utilities on IF in Medellin, Scopus, PubMed, and Google Scholars. Out of our research, we found 439 that have anything related to cost in IF. Then we brought it down to 297, and after apply our eligibility, we end up with 37, and out of that we excluded 31, and the final results was 6 articles that met our inclusion criteria. So 2 of them use cost analysis only, 1 use cost benefit analysis only, and 2 use cost analysis and cost effectiveness. And the last one just use both cost benefits analysis and the cost utilities. So the conclusion of this study was that individuals actually value the returns of IF, and they are actually willing to pay for them. Also, the cost of returns IF depends on the primary health conditions of the patient. We also found that there is no clear evidence that returns IF might be cost effective. So now that brings us to the next study, which is the cost of returning Africa recommended IF to research participants. So as you can see, there is not a lot of out there that provides the clear picture of the cost, what the cost of returns IF. So our objective was to develop a quantitative model to assess the cost of returns IF in a research setting. So the cost analysis tracks participant level costs in the FXS project. So the method was, we explored how return results to participant impact the research budget for cascade testing for the FXS project in Cameroon. So the cost analysis basically, well, as I say, tracks the level cost of this project. So we separated into 4 different steps. We have the first step, which is identifying a recurring sample. This means like going to community, explaining what is the research about. Maybe they had to come give some gifts to the community, and just have people involved in this project. The second part of the step is to the DNA sequencing and the variant interpretation. So actually take the DNA of the participants and doing the sequencing analysis. The third step is the feedback to participant. And the fourth step would be the medical care and the follow-up up to IF returns. So for this research, the FX project in Cameroon, they have a total of 46 participants. So at this point, this project haven't done IF's analysis. So what we did was we estimated the numbers of IF prevalence using the literature, which is on average 1 to 3% of the size of the study cohort. So therefore, 46 participants would give at least 1 participant that would have IF return in this case. So here's our result, the summary of tables that provide the first column provides the results of the project, the actual results of the project with the cost. And then the second column would be the cost, let's say, including this one, if the research would be willing to return the IF, what would that cost to the project? So, and then the third column would give us the difference. We'll see that the difference in including the IF would be around $1,947 in this case. Which bring us back to our discussion in conclusion. The returns shows that the returns IF to participant will initially have a high cost to the project. However, the cost of returns IF to participant might decrease if more participants are enrolled in the project. That said, that you notice that the cost it's higher in the beginning because you have to do the consent and all this translation. You have to have another let's say gathering with the participants just to talk about what IFs are and counseling and stuff like that. So that means that the cost right there would be much higher. That's like goes to my next point. To most of the cost of research was related to building a trust relationship with the community. The lowest cost of the research was related to step 4, which is the follow-up of the participant. However, this cost does not include the cost of acting on it. We can see that depends on what IF and what the participants would act on it. It can vary depend of the disease. So yeah, that's about it. Thank you very much for listening. And if you have any questions, please let me know. I'm willing to answer any questions that you have. And I really appreciate to be involved in this project. And yeah.
Thank you very much. We're going to pause now for a Q&A session. Do we have all our panelists available? We do have a question for Arisa. Let me just get it on the screen. And this concerns the issues about knowledge of genetics. And there's a question as to whether nurses are more or less knowledgeable, or is it possible to say anything about this on genetics than the general public or other healthcare professionals such as doctors?
Okay, thanks very much. I hope everybody hears me well. About the comparison of the level of knowledge amongst nurses compared to the general public. Now, this is information we did not collect for our study. However, there is another ongoing study in Uganda, still on genomic and genetic research that has gone to the community, to the grassroots, and maybe at the end of that study we might be able to answer that question. Apart from the MDs, yes, we recruited only researchers conducting genetics or genomic research. So basically, for them, they were knowledgeable because that's their field of research. Thank you.
Thank you, Arisa. Well, the next question was addressed directly to Dimfo, but I don't see Dimfo on the panel. But there is Dimfo. Right. Thank you. You've come into my vision now. And the question is, what reasons do we know about for why some participants don't want to receive feedback regarding the predisposition to severe disease? Do they give a reason why they don't want to get return of results?
Thank you, Chair. Actually, we found that most of the people who were reluctant to receive findings that were severe, not preventable, and unactionable were the adolescents. And their reasons was that there could be psychological implications. They said that knowing such results is not useful because there's nothing that they can do about such findings. So there's no need for them to know about that.
Thank you. I'm just wondering whether Mayara might also want to comment on this question, given what you were saying about how people were willing to pay for results in some circumstances.
Sure. Actually, we're looking at literature, and most people they actually afraid of receiving the IFs because they don't know. It's like learning what could happen to you. It brings fears to them. So we saw that in the literature. That's why most they don't want to know that they have predisposition for cancer, and they're afraid that they're going to put that burden of cost to their families. So that's one of the reasons why I seen it. Yes.
Okay. Right. There's another question for you, Mayara, which is what currency are the incidental findings, costs, prices in your tables? And
it's US dollars. Sorry, I should have mentioned that. We converted to 2016. Yes, it was converted.
Okay, and is the cost just giving the results or including the intervention? I think you did say something about the different types of cost towards the end, but the question goes on to ask how many times is that cost above the national per capita allocation per person by government?
Oh, that's a good question. I haven't looked into that, but I'm going to take that note. What I also looked into was what would be the cost of acting to diseases that are predisposing in the African population. So it could potentially spend on average, like let's say if a person gets IF results, depend of course of the disease, it could potentially spend between $21 to $250,000, or they could also save $53 to $30,000. Also depends of the disease. And these diseases are usually related to, let me just see, guys, sickle cell disease. I broke it down by also G6 PD deficiency, breast cancer, kidney disease, and yeah.
Okay, well, a follow-up on that. I'm not quite sure I entirely get the full meaning of the question, but it's from Colette Dandara, and it says, is African recommended supposed to also mean African specific recommendations? And would it lose meaning if they were just referred to as recommendations regarding the return of IFs?
Okay, so if I understand correctly, the question is if Africa research recommending the returns of IF to participants. Is that correct?
I believe so. The question is asking about whether they are African, the recommendations are African specific.
They are actually the H3 Africa actually is creating a list that contains 3 tiers of reportable findings based on evidence, and I don't think they haven't published yet. I actually introduced that in this analysis, but this analysis hasn't been published yet. We are finishing it up and we're going to submit it soon. But there is a list that they are recommending for African specifically.
Okay. Thank you very much. Well, I think that's very interesting exchange. We can now, I think, move on to the next set of presentations, but thank you for the presenters so far.
Thank you.
So now we have 2 presentations on the TB Gen project. First, Jonas Hirutu. Over to you.
Hello, my name is Jonas. I am presenting on progress and genome sequencing updates of TB Gen project. As an introduction, tuberculosis is caused by Mycobacterium tuberculosis bacterium and is a major health problem in Africa. TB Gen is a research initiative engaged in host and pathogen genetic variation and its impact on disease outcome. TB Gen is a consortium project of 4 African countries, Ethiopia, Eritrea, Sudan, and Cameroon. The project involves a cross-cutting fields on bioinformatics, immunology, and ELC research and capacity building at all levels. The major objective of the project is mapping the genetic and geographic structure of the bacterium, testing the hypothesis on the overlaps of the genetic and geographic structure of the host and the pathogen. And third, to investigate the correlation between host and pathogen genome with a distinct clinical outcome. The project also has a major objective in capacity building for genomics and bioinformatics. In order to address this research question, a sample size of 2,200 paired samples will be collected from 4 countries. So far, 956 paired samples are collected. Whole genome sequencing of 55 MTB isolates are ready for analysis. Bioinformatics pipeline developed for MTB, and the work is in progress for human genetics. In terms of capacity building, 2 MSc students graduated, 9 postgraduates, and 6 research fellows are currently engaged in genetics, immunology, and LC fields. 2 grants and scholarship won by the team member. Senior bioinformaticians have also joined the team. The team also hosts 2 training organized by H3 Bionet. For 5 MTB clinical isolates sequenced in 2020, the bioinformatics team conducted a pan-genome analysis. The bioinformatics pipeline followed an input from Illumina read, quality control, de novo assembly, and variant mapping. From this data, in silico genotyping, from the assembly data, virulence factor, T-cell epitope mapping and identification was conducted for a comparative pangenome analysis. The results include functional annotation of assembled sequences for comparative pangenomic and highlighted the generality and the strain specificity of the strains compared to the reference genome used in the study. Secondly, also, the genetic diversity of the isolates were also compared. One example showed here a phylogenetic placement of the strains among representative lineages of MTB. In order to address the diversity of virulence factors, all experimentally proven and putative MTB virulence factor genes were obtained from the database. And a series of blood searches were conducted, non-synonymous SNP distribution were mapped. As shown in the Venn diagram, a common and individual distribution of non-synonymous distribution were mapped. Secondly, in order to identify lineage specificity of this variant, a large-scale previous works were used as a reference to find an overlap and found an interesting result for lineage 3 and lineage 4 strains of the clinical strains of MTB used in the study. Further filtering was conducted in order to identify only promising non-synonymous SNPs. Phylogenetically informative barcode SNPs were masked by comparing an overlap with 1,600 MTB global strains of MTB. Secondly, only functionally relevant non-synonymous SNPs were considered. From the table, interesting results were obtained and presented here, for example, for LC strains with deleterious non-synonymous SNPs for virulent factor classes of secretion system and functionally classified and involved in cell wall and processes were more pronounced. Similar strategies were followed for identification and mapping of T-cell epitope and compare with the reference genome from virulent and non-virulent strains of H37. As expected, 80% of T-cell epitopes were hyper-conserved, and from the accessory T-cell epitope, heat map were generated and show clustering among the clinical strain and compare them with the reference genomes. Further comparative analysis was conducted to identify variants on mutated T-cell epitope. The table showed the number of mutated T-cell epitopes shared among the clinical strains that are in common and strain specific. Interesting to note here that the number of mutated T-cell epitope between lineage 3 and lineage 4 strains were different. And this was inconsistent with previous reports that L4 are generalist and more hyper-conservation of T cell epitopes were expected. And this could has implication on host adaptation and success for global transmission of L4 strains. Taken together, a draft manuscript is prepared and it's under review. Finally, on human genetics work, TBGene project established collaboration with H3 Bionet to undertake whole genome sequencing and array genotyping on subset of representative human samples based on the genetic background, disease severity, comorbidity, lineage of MTB, and other clinical lab tests. And this will be the first host genetic variation data that will be compared to the genetic diversity of the pathogen and further establish targeted enrichment sequencing assay. The future plan includes finishing sample collection and processing, data generation and analysis, and further strengths capacity building. And finally, we'd like to acknowledge our funders, partners in institution, and study participants. Thank you.
Thank you very much, Jonas. We'll go straight on now to Nchangwi Cynthia, who's going to talk about challenges of informed consent in this project.
Good day to everyone, and thank you for the opportunity to do this presentation on informed consent, comprehension and recall of study information by study participants of the Tuberculosis Genetics in Africa project at the Cameroon site. I'm doing this presentation on behalf of Mr. Ali Ibrahim, who is of the TBGen Eritrea site. He conducted the study in Cameroon because he was registered at the University of Buea in Cameroon. On the slide are the different supervisors, and I'm one of his supervisors. Ali has since returned to Eritrea upon completion of his master's degree, and he has limited internet connectivity at this time, so I am presenting on his behalf. So Ali had set out in his master's thesis to assess consent comprehension and recall of information by participants who were enrolled in the TBGen Africa study at the Cameroon site. So this included identifying factors that motivated participation in the TBGen study and exploring factors that could influence comprehension and decision making. This was a qualitative research study. Ali enrolled participants from the southwest region of Cameroon, mainly Buea and Limbe, and these are the English-speaking regions of the country. In terms of the study participants, this table gives an idea of the participants that were enrolled in terms of gender, age group, and educational status. But just to highlight here that the majority of participants for this study were very educated, and they had at least high school education. For the results that Ali got, in terms of understanding of genetics, which was a key question that he posed to research participants, generally participants understood genetics and explained it in terms of hereditary, and that was really great. But in a way, it affected the way participants understood the objectives of the TBGen study. And this is mainly because participants argued that TB, so tuberculosis, is not hereditary, and therefore it wasn't possible that you would have a genetic study on tuberculosis. Only one participant explained the study objectives in terms of susceptibility, and that participant explained that in a previous experience, a medical doctor had explained to him issues around TB susceptibility and your environment. But overall, all participants did not understand the objectives of TBGen and argued that it wasn't possible that it was a genetic study. Participants also had limited recall of the biobanking and data sharing aspect of TBGen. And I think the key message here is that there is a need to seek ways of explaining genetic susceptibility to participants. And this is really important for genetic studies that are focused on infectious diseases. In terms of overall perception of their participation in the TBGen studies, the participants were quite optimistic and were very happy to participate. And they gave 3 main reasons for why they would participate in the TBGen study. And the first was that they thought there was no risk, because if there was, the medical doctors would have explained that to them. Worthy of note here is that participants for the TBGen Cameroon site are recruited at the TB clinic at the different regional hospitals. Another reason why they were really positive about participation in TBGen was because they wanted to help the postgraduate students that were enrolled at the University of Buea. So Buea is really an education city, and the main activity in that city is the university. And so it's quite common to see, so the population is really sort of familiar with the activities of the university in terms of research. And another reason, which could be described as diagnostic misconception, is that they participated in TBGen not knowing that it was research. So they actually thought that it was a way of getting more advanced or more knowledge about their disease or more advanced diagnosis. In terms of the overall attitude to biobanking and data sharing, participants were generally, so there were heterogeneous views around biobanking and data sharing. And while the participants were had an overall positive attitude, their motivation there was still related to diagnostic misconception. They thought that biobanking and data sharing was important, not because of research, but because they thought if their samples were stored and sent to Ethiopia, which is the hub of the TBGen project, then they would be able to know more about their disease in terms of diagnosis. The factors that affected, so participants had very limited, could recall very little about the study information provided to them during the consent process. And firstly, also it is because of diagnostic misconception. They never thought, many of them did not know that it was research. Or they joined the study because they thought that it was about access to diagnosis. And so the rest of the study information, they didn't pay attention to that. There was also the issue of information overload, in that at the time of enrollment, there were other studies that were going on and which were recruiting the same participants. So at some point, participants had to sign like 2 or more consent forms. And so they thought that that was too much. They couldn't remember, because at some point, a participant, some of the participants for this study said the study was about filaria or about schistosomiasis, but that was because there were ongoing studies at the time on those different diseases, and participants had also consented for that. So there was some sort of information overload there for participants. And then there was the issue of situational vulnerability, in that many of the participants that were enrolled had just received a positive diagnosis for tuberculosis. And so when they were approached to join the TB-Gen study, many of them said they accepted because they just wanted the researchers to leave them alone at that time. So they thought a yes would be a best way for the researchers to go. And so the key point there is that we should sort of give time between the time we enroll for the TB-Gen study and the time that participants receive a TB positive diagnosis, just to give time for them to assimilate the information they have been provided in terms of diagnosis and when we introduce the study to them. And also that the simultaneous consents for different projects, even if they are related, may be confusing to participants. So Ali would like to acknowledge quite a number of people, including his supervisors and the TB-Gen team in Cameroon, but he really wants to say thank you to the TB-Gen field staff for helping him with the interviews, especially in Pidgin, which he is not familiar with, but which is a language that is commonly spoken in the English-speaking part of Cameroon. Thank you for listening.
Thank you very much. That was a most interesting case study about the challenges of informed consent, especially what you said about susceptibility. But we're going to go straight on. I'd just like to remind attendees they can still post questions about that session for the Q&A at the end, but we're going to go straight on to the next talk, which is Derek Wildman, and it's going to be a talk about epigenetics in relation to the Rwandan genocide. Thank you, Derek.
And thanks to everyone attending and to the other speakers. It's been a very interesting session. I am going to talk about epigenetics in relation to the Rwandan genocide against ethnic Tutsi, some pilot work that our team has been doing. The team is led by Dr. Leon Muteza of the University of Rwanda, Stefan Janssen, Monica Udine, and myself, but there are many other people working on the project, and we'll hear about them in a little bit. So I am going to talk about the pilot epigenome-wide association study, but I will also give an overview of some of the things that have happened that have slowed up our project a little bit as you're all aware about COVID and stuff like that, and some of the other accomplishments our team has made. And so I will just dive right in because I know we're running a little bit late. So there's Rwanda, as I'm sure all of you are aware where it is. This study is being done to get a sense of what happened epigenetically in relation to the 1994 genocide in Rwanda. And that between April and mid-July 1994, hundreds of thousands, if not over 1 million people were killed. It was a big, terrible situation. 1 in 4 children in the country were orphaned. And we study post-traumatic stress disorder and trauma, and amongst women and orphans, about 41% of folks have been diagnosed with PTSD. This is just a picture of the genocide memorial in Kigali. This is a group burial also there. So the overall goal of our research was to characterize the transgenerational genomic impact in genocide exposure and PTSD in women and survivors of the Rwandan genocide, as well as their offspring. We are particularly focusing on women who were pregnant during the genocide. I want to just say a little something about COVID because that's affected our research. These are some stats that show the COVID situation in Rwanda as of last week. And I will just say that Dr. Muteza is one of the leaders of the COVID task force in Rwanda. A PhD student on our project also is highly involved in that COVID response, as are other members of our team. And Dr. Muteza and Dr. Uwaneza, who's pictured in the middle here, are the only clinical geneticists in the country. And so their role is very important, and their student Clarice is shown on the left. They've been working hard, and this is showing a hypergeometric pooling regime that they've developed to facilitate the testing of samples of people who've been exposed to the virus. And that's a really important finding because of the cost savings of pooling samples compared to the way it's done in the United States, for example. Back to PTSD. So 28% of genocide survivors have been linked with post-traumatic stress disorder, which is higher than you see in other places. PTSD globally is about 3 to 4% of folks. So there's been a big burden on the population there. So in our pilots, in our full study, we're looking at 300 samples, mothers, offspring, and younger siblings, and then people who were outside of Rwanda during the genocide but are ethnically Tutsi. So all of the women were pregnant during the genocide. And the offspring who were in utero during the genocide and subsequent offspring are included in our study. A lot of the participants come from the Association of Genocide Widows, Agahozo, which was established in 1995, shortly after the genocide, to reintegrate genocide widows back into Rwandan society. There's about 20,000 plus members in this group. We also found participants at a place called Compassion International, which is a faith-based organization that focuses on the health and well-being of children. And this is some of our study participants. This is a picture taken at Avega, and these women are involved in the study, and this was part of our community outreach efforts. So what we're doing is we're determining DNA methylation levels in participants exposed versus unexposed, and identifying epigenetic differences associated with the intergenerational transmission of trauma and PTSD. So we've already seen that. In our pilot study, we just looked at 70 participants, 31 cases and 39 controls. And the technology we used was something called the Illumina Infinium Methylation Epic Bead Chip technology. And this is our pipeline, and this was developed by Clarice and Agaz, Dr. Wani and Miss Musan Baganawa. And the purpose of looking at these samples is to determine non-mutational changes, epigenetic changes that occurred in these participants. And we looked at DMRs, which are known as differentially methylated regions, which combine several CPG sites. This is a list of genes that were found to be significant, meaning that the adjusted p-values using the Benjamini-Hochberg method were less than 0.05. This is in comparing genes that are differentially methylated in children exposed versus unexposed to the genocide. So these are kids who were in utero in 1994. And you can see there are some significant hits. Then ultimately, we're interested in intergenerational or transgenerational inheritance of epigenetic marks. And so these are significant DMR genes in both the mother and the children. And you can see some of them are involved in development, like the HOX gene there, involved in a variety of processes and functions. Focusing on blood is the way our study's done, because it's not possible to sample brain tissue, but we can compare genes that are known to be methylated in similar ways in both blood and brain from other data sets that are available. And so we can see these three here are differentially methylated in leukocytes and in central nervous tissue. And so one of these is B-core, and it's significantly differentially methylated in both the children and the mothers. The genocide exposure, therefore, is associated with DNA methylation in this gene B-core. So these are the three that are in the red boxes that have a brain-blood similarity in terms of methylation patterns, and are particularly interesting candidates for us, and we'll be looking at those genes in more depth as we expand our sample size. So many DMRs were found to be significant in children and mothers. 3 shared DMRs showed significant correlation in DNA methylation in brain and blood tissue, and statistical evidence for intergenerational transmission possibly was identified in those 3 DMRs. Now, so data collection in Rwanda paused after the pandemic began because of lockdowns and such. Our labs have been active throughout, and we were fortunate to have samples from 47 families sent to our university in Florida in January of this year. The DNA from these samples is of high quality. We've put them on chips, both the H3 Africa chip and the EPIC bead chip that I mentioned previously. So that allows us to look at both genotype and epigenotype. And as you all are aware, that the Illumina chip, the H3 Africa chip, is designed to capture African genetic diversity, which is something that hopefully will improve the understanding of diversity on the continent. So far, we tested 141 samples on the EPIC chip. 138 of those survived QC metrics, and when all was said and done, we were able to use 135 of those samples, and we can examine methylation at 820,000 approximate sites. We've also been working on other projects. This is a recent meta-analysis that Clarice led, and what it's showing is that the prevalence of PTSD is higher in genocide survivors. We've also been involved in the mental health task force. That is an effort that is meant to harmonize across studies that look at mental health outcomes in the consortium. And so the 3 main ones we've been working with are ours, and then the Drakenstein Child Health Study, and the DDD Africa study from the DRC and South Africa. And So we're working on harmonizing this with the teams. This is just a slide that shows how it's challenging because the studies don't all collect the same type of data, but the green parts are things that we can compare across a number of studies. We've also been involved in the publication working group in the consortium. COVID work in Rwanda has been very, very positive for the most part, certainly better than what we see in Florida. One of our team members, Shagun Fatumo, is now started as a postdoc on the project, and now he's an assistant professor in London, and he recently got a very nice grant from Wellcome, and we're learning from him a lot. He was written up in Science earlier this year about his sort of path to becoming a genomicist, and Shagun is an expert on GWAS and cardiovascular disease and many other things. We've been going back and forth as best we can, despite the travel disruptions. You know, Dr. Udine went to Rwanda a few times. Dr. Fatumo came to our genomics program at USF for several months to learn some of the techniques that we use. I've gone to Rwanda to help with the optimizing of the DNA isolation protocols and the serum and stuff like that. And then Clarice came to USF also and learned a lot about high performance computing techniques and so on. The summary of our project is that there's evidence for intergenerational transmission. Yep, that's fine. I'm pretty much done. I just want to thank all our partners who work on this project, and thank H3 Africa and everybody else. Thank you for your attention. That's it.
Thank you very much for that fascinating talk, Derek. We need to go straight on because we're running a little bit late. Our final two talks, the first one is by Patricia Lewandwe. Over to you.
Greetings, everyone. My name is Patricia Nevonde. I am a PhD candidate at VET, and today I'll be presenting a part of my PhD project. Developmental delay is an umbrella word which describes conditions in which children exhibit a significant lag in attaining desired milestone for their particular age in at least one major domain of development. The major domains of development include gross and fine motor, speech and language, cognition and perceptual, as well as personal and social development. Developmental delay is caused by a combination of environmental and genetic factors, with environmental factors including maternal alcohol consumption and as well as gestational infection. Studies looking into the genetics of developmental delay have identified causal mutation in 13 to 60% of the cases, with approximately 42% of cases harboring de novo mutations. And some of those de novo mutations identified are located in DNM-enriched genes. De novo mutation-enriched genes are genes who show an excess of de novo mutations as compared to the expected number of de novo mutations generated from predicted mutation rates. These genes are highly involved in critical developmental pathways such as chromatin remodeling, cell proliferation, and neuronal migration. These genes are mostly intolerant to variation, thus the majority of mutations within these genes could be damaging and are more likely to influence developmental delay. And this makes us question if targeting mutation within de novo mutation-enriched genes can be an attractive diagnostic approach for developmental delay in resource-limited settings. And this brings me to the aims of my study, which are to investigate the contribution of de novo mutation-enriched genes to the genetic etiology of developmental delay in South African patients, and to evaluate the utility of a developmental delay diagnostic strategy based on de novo mutation-enriched genes. Now, moving on to the methods of this study, a comprehensive review of the literature was carried out in order to identify the candidate de novo mutation-enriched genes to include on the virtual gene panel. The virtual gene panel was then designed and analyzed in 96 data samples of patients with developmental delay. These are the patients who had underwent whole exome sequencing as part of the DDD Africa project. These data samples had already went through pre-processing quality control filter and variant calling. Variants were then annotated using the variant effect predictor tool. And when it comes to variant filtering, we, number one, firstly filtered out all synonymous variants, and then we filtered out variants with a minor allele frequency of more than 5%. We then prioritized variants based on various bioinformatics prediction tools. Variants were then classified and interpreted following the American College of Medical Genetics guidelines. Based on these guidelines, variants were classified into 5 groups. Thus, pathogenic, likely pathogenic, variant of unknown significance, likely benign, and as well as benign. All putative disease-causing variants, those are the variants which are classified as pathogenic or likely pathogenic, were then visualized on the integrative genomic viewer. This was done for a number of reasons. Number one, to confirm that the variant is a true variant and not just a sequencing or a calling artifact. And number two, to note the allelic fraction between the reference and the alternative allele at the variant's genomic region. This was done as part of the post-processing quality control. All variants which passed post-processing quality control are currently being correlated with the clinical phenotype of each patient by a multidisciplinary team. Now, moving on to results, a total of 285 de novo mutation-enriched genes was identified and analyzed in 96 patients. After our primary data analysis, we identified 110 putative disease-causing variants in 66 patients. Following our post-processing quality control, we were then left with 44 putative disease-causing variants in 37 patients. These are the variants which are currently being correlated with the clinical phenotype of the patient. And I am now going to present to you the result of the first 3 patients whom we have completed phenotype correlation. Patient 1 presented with moderate intellectual disability, dysmorphic features, relative macrocephaly, short stature. This patient was negative for karyotyping and MRPA. Based on the patient's clinical phenotype, the patient was suspected to have Cornelia de Lange. After data analysis, we identified pathogenic variant in the ARID1B gene, and thus the molecular diagnosis of this patient is Coffin series. Patient 2 presented with mild to moderate intellectual disability and dysmorphic features. Like patient 1, this patient was negative for both karyotype and MRPA. Based on their clinical presentation, this patient was suspected to have Coffin-Lowry or Coffin series syndrome. And after data analysis, we identified a pathogenic variant in the PECX1 gene. And thus the molecular diagnosis of this patient is PECS1 syndrome. Our third patient presented with moderate to profound intellectual disability and was negative for both karyotyping and MRPA. This patient had no suspected diagnosis, and after analyzing data, we identified pathogenic variant in the CHD7 gene, and thus the molecular diagnosis of this patient is CHARGE syndrome. Moving on to discussion. Our preliminary results suggest that a de novo mutation-enriched gene panel has the ability to discover novel findings in African population, and thus it can expand on clinical phenotypes and molecular profiles of patients with developmental delay. Additionally, it has the ability to end a diagnostic odyssey by providing a positive result. There are numerous benefits of a positive result, and these include the ability to now offer accurate genetic counseling. A positive result can direct a person towards available prevention, monitoring, and treatment option. Furthermore, it has been reported that individuals who undergo genetic testing have reported showing interest in receiving results for personal utility reasons. These reasons include the ability to take well-informed reproductive decision-making, enhancing their self-knowledge, and as well as identifying useful support and advocacy group. In summary, our preliminary results suggest that a de novo mutation enriched gene panel could be a useful diagnostic approach for developmental delay in resource-limited settings. This study is still in progress. Future work will include correlating all identified putative disease-causing variants with the clinical phenotype of each patient, feedbacking pertinent findings to the patient, as well as determining the overall diagnostic yield of de novo mutation-enriched genes in this cohort. I would like to acknowledge the following people and funders. This brings me to the end of my presentation. Thank you so much for listening. I will now take questions.
Thank you very much. We're now going to go to our last speaker, but before we do that, I'd just like to say that we're not going to have time for a live Q&A, but all the questions you can look at in the Q&A box, and some of them will have been answered in the box, and you can find the answers if you press on the button Answered Questions. Okay, thank you very much. So let's go straight on to the last speaker.
Thank you, the organizing committee, for this opportunity to present part of my work, which is of the DDD Africa. I am Jerome Obongo. I am a pediatrician and a clinical geneticist from the University of Kinshasa. My research study is entitled Objective Evaluation of Dysmorphism by Automated Analysis of Facial Photographs. This is the outline of my presentation. Dysmorphism, meaning the presence of 3 or more minor anomalies, has a role in defining prognosis. It's a robust indicator of an increased risk of a major anomaly. Dysmorphism increases the chance of detecting a pathogenic copy number variation. One of the difficulties in evaluating dysmorphism is that this might be subjective. There is a growing need for more objective evaluation. Here, we explore the automated evaluation of facial dysmorphism using FACE2Gene. The general aim of this study was to evaluate the use of automated analysis of 2D facial pictures. To achieve this aim, we assess the accuracy of FACE2Gene to recognize distinct minor facial anomalies as well as facial dysmorphism. And we explore the effect of gender, age, and ethnic background on the facial phenotype. We have included 4 cohorts in this study. The pictures of 481 newborn cases from the previous study, of 126 children with developmental disorder, of 17 children with Down syndrome, and of 21 healthy volunteers were uploaded in FACE2Gene for further analysis. FACE2Gene uses an algorithm to evaluate facial features. It uses 2 tools, the clinical and the research tool. The clinical tool suggests a list of facial features observed in the top matching syndrome. In addition, it returns a list of matched syndrome ranked by their probabilities. In the research tool, one can compare facial photographs of different cohorts to see how well different or similar they are. The output is given in 2 ways. The score distribution showing visually whether the 2 cohorts are separated, and the work with the IUC as a measure for how well cases from the 2 cohorts can be discriminated. In this example, the IUC is 0.981, which is very high, meaning that the faces are indeed different in the 2 cohorts. The first question was, Can Face2Gene extract accurately facial features from the pictures? In clinical setting, morphological features are divided into two types. Common features are those observed with a frequency of 4% or more in the population, where rare variants considered as minor anomalies are those observed in less than 4%. Face2Gene identified more morphological features than the clinical evaluation, and those features had very high frequencies as compared to the clinical evaluation. We concluded that the threshold of 4% was not applicable to the automated evaluation. Instead, we used a threshold of 10% to separate between common variants when equal or above 10% and minor anomalies when below 10%. The second question was whether Face2Gene can be used to define facial dysmorphism. We compare, therefore, our clinical assessment of dysmorphism with that of Face2Gene. In the x-axis, we classified newborn in 4 categories regard to the number of minor anomalies after clinical evaluation. And then in the y-axis, the percentage of those that are dysmorphic as scored by Face2Gene. As shown here, dysmorphism is defined as 3 or more minor anomalies. You can see in this cohort with a low incidence of dysmorphism, the specificity was 94% and the sensitivity poor. Nevertheless, this is promising to screen for dysmorphism if one accepts a low sensitivity. Using Face2Gene research, we first evaluated ethnicity. Here, we compared children with Down syndrome from different countries. There is a clear difference between Africans and Caucasian and a significant similarity between African in Africa and those overseas. The phenotypic distances are reflecting the genetic distances. In our previous clinical study, we evaluated differences in incidence of common minor features between newborn from different ethnicities, but did not find significant differences. Likewise, Face2Gene was not able to separate newborn based on the ethnic group. We also evaluated sex. Face2Gene was not able to separate newborn and young children below 10 years, but obviously, adults could be separated with regard to sex. It's likely that the incorporation of sex, ethnic background, and age in the algorithm will improve the performance of Face2Gene. In this study, we established the feasibility of dysmorphism evaluation using automated tool. We have provided evidences of the influence of age, gender, and ethnicity on automated dysmorphic evaluation. By doing so, our study has established the framework for objective evaluation of dysmorphism in African descent. We sincerely thank the team that contributed to this project. Thank you for your attention.
Thank you very much. Well, that talk brings the session to a close. I'd like to thank all the speakers and attendees. It's been a most interesting session. I'm sorry we haven't got time for a live Q&A, but we have another plenary session coming up. So thank you once again.